So I'm now going to call up Dr. Marek Radomski, who is with the University of Saskatchewan, to bring greetings and introduce your speaker.
Thank you very much, Michelle. It's a real pleasure, guys, to see you all here. I represent the University of Saskatchewan and College of Medicine, and I hope that some of you may join us in the campus in not too distant future. Become the student scientists. Will be absolutely thrilled and delighted. My great task today is to introduce an extremely accomplished and successful fellow scientist, Dr. Roel Nusse. Dr. Roel Nusse is native of Netherlands, and he got his undergraduate degree, Bachelor of Science, and then the doctorate at University of Amsterdam in Netherlands. And then he chose to pursue his career in the United States. He moved to California, to a great place, University of California in San Francisco, to work with the, at time, already one of the best cancer scientists in the world, Dr. Harold Varmus. And then a few years ago, it was I think it was 1982, if I am not mistaken, Roel and Harold Varmus made this amazing discovery about the WNT signaling pathway, genes and proteins that actually control the way how the signals, how the chemicals from outside the cells get into the cell and what would be the repercussions of this entry. And the repercussions are of extreme importance. It's the development of the embryo. This is how we start growing. If the things go wrong, we may develop the cancer. And also the part of the current activity of Dr. Nusse lab is about stem cells and the tissue regeneration. And the common denominator with this is WNT. If you look at the citation for the Gairdner Award that Dr. Nusse received in 2020, it reads, for pioneering work on the WNT signaling pathway and its importance in development, cancer, and stem cells. Dr. Nusse is a professor and chair of the Department of Developmental Biology. He is a fellow of the Institute for Stem Cell Biology and Regenerative Medicine at Stanford University School of Medicine. He is also Virginia and Daniel Ludwig Professor of Cancer Research, and last but not least, the researcher at the Howard Hughes Medical Institute. So you've got and I have this great pleasure and privilege to introduce to you one of the best biomedical scientists that Gairdner's Foundation awarded with our Canadian award. Noel, the floor is yours.
I have a microphone. Yeah. So thank you very much, Marek, for this introduction. Also for hosting me. And I also want to thank you guys for attending this lecture. I'm looking forward to giving you an overview of the fundamental background behind our research. I do a lot of teaching at Stanford, and I'm used to students interrupting me during the lecture. So if you guys have a question about the slides or what I'm saying, then please raise your hand and ask me a question. So the topic of my talk is research into the links between embryogenesis and cancer and how cells influence each other. And to start the lecture, I want to point out that this is already a long time ago that a scientist, his name is Rudolf Virchow, he became interested in the roles of cells in disease, and he was able to draw these beautiful diagrams of cells that he could detect under the microscope. And not only that, he was one of the first people to recognize the importance of cells in tissues, he also postulated that many diseases were the consequences of loss of cells, and we now realize that that is indeed the case. He also suggested at the time that cancer, now this was in 1855, there's almost nothing known about cancer, was due to the overproduction of these cells. And we now, of course, know that our bodies and organs, they do consist of cells. In the lung, we have cells of the lung. The bone, surprisingly, contains all kinds of cell types and other materials. And then we have our immune cells. They recognize and they kill strange cells, and they're present in the blood to detect invaders. And of course, there's many other cell types in bodies. Now, more recently, scientists have been able to watch and visualize cells with very advanced microscopes. And later in my talk, I'm going to use some of these microscopes actually to show you the pictures of cells. And what always fascinates me here is that, so these are living cells that are in the dish, and they have been marked with a protein. It's called GFP. You probably have heard about it, that actually is able to visualize certain proteins in cells, and in this case, proteins present between membranes in cells. And you can see that the cells adhere to each other while they're actually migrating in the dish. Now, there was more that Virchow postulated. He suggested, now this is in Latin, that omnis cellula acellula. And that means that all cells actually arise from pre-existing cells. And here, a simple diagram, he was also right. What he noticed is indeed, under the microscope, and I'll come to show you one example, is that cells arise from pre-existing cells. This is an embryo. It is an embryo of a worm called C. elegans. And what you're going to see now is that the cells are going to divide. You're probably all familiar with it. I added some music to make it more dramatic. You see that these embryos, they start of course as one, and then they become two cells. They quickly turn into four cells. The cells start to move around, and are later going to form the different organs of the C. elegans embryo. Now it starts again. So Virchow was right, and he also was one of the first people to recognize that when cells divide, the chromosomes that carry the genetic material, they have to duplicate, and they have to become distributed over the daughter cells. And this is essential, of course, when cells are going to divide for more cells, that each of the cells is going to inherit the genetic material of the mother cell. And that is visualized here. Chromosomes that are going to adhere to what's called the metaphase plate. And then the chromosomes are going to be pulled apart so that the daughter cells are going to inherit the correct number of chromosomes. These are fundamental processes, and you guys have to realize that even though this has been seen for many, many years, we still don't understand the details of how chromosomes become partitioned over daughter cells as they are dividing. And this, of course, is also instrumental in thinking about incorrect ways that chromosomes are segregating that may also have consequences for the origin of cancer cells, as often cancer cells don't have the correct number of chromosomes. So, yes, most cells can divide and make more cells. However, most cells in our bodies do not divide. This is also very important to realize. Most cells are specialized and they execute the function of the tissue. I showed you this example here of a cell in the lung. There's a cell in a structure in the lung called the alveoli. This is a cell that is involved in the oxygen uptake. And this is a non-dividing cell. It has a limited lifespan as well. It is unable to proliferate as many of our cells in the skin, in the bone, in the liver, and the organ. Now what happens over time, when cells are executing their function, they start to die. And this is in the whole thinking about turnover of tissues and also the generation of cancer cells, something that is very important to realize. So the mature cells, the specialized cells in our body, on the one hand, they cannot divide. They die by something that is programmed cell death. And that process of programmed cell death, that involves the fragmentation of the membrane surrounding the cell and also the nucleus. And there are staggering numbers of cells in our body that on a daily basis, they die. People estimate that between 50 and 70 billion cells die every day due to that process of programmed cell death. So more examples are cells in the blood. The red blood cells that you're all familiar with, they have a lifespan of about 6 days. So they have to be replenished, right? That is very essential. And this is now an example of cells in the petri dish. They start off being alive, they move around, but then some of them are going to die. You can visualize it here. The music now is kind of sad, because all these cells are going to expire. Here's another example. This cell is going to die, the other one as well, but this one I think is going to be first. This one. So it's a very intricate process, right? That these cells, they start off having a nice membrane and suddenly the membrane explodes and the cell is dead. We'll watch it one more time. Single cell. This is the nucleus of the cell now. And now it's dead. So, now I'm going to talk about the process of tissue renewal. We've now understood that the specialized cells in the body, and these again are all the cells that you're so familiar with, cells in the lung, in the skin, in the bone, they do not proliferate, they do not divide, but they die. So then the question is, and this is essential for this whole topic of stem cell research, how are the lost cells in our body being replaced? What is the source of new cells once your existing cells have died? And this is now where stem cells come in. It turns out that in almost any tissue that we know, there are a few cells that are replication competent. They do not die. They can renew themselves and generate the specialized cells. So they proliferate, they make more stem cells, but they also start to make specialized cells. Now, it also turns out that these specialized cells, these stem cells, are regulated by short-range signals. The signals coming from other cells that influence the behavior of the stem cells. And these signals are very essential for the behavior and the proliferation of the stem cells in our body. Again, they're replication competent, but they only start to divide, they only start to replicate once they receive an appropriate signal. And here's another example of a tissue. This is now the intestine, the tissue, the gut, where there are a number of differentiated, specialized cells. They form the villi that stick into the lumen of the gut. These cells are also going to die ultimately, and it has been found that these cells are going to be replaced by stem cells that are in a different location in the tissue, down here in the tissue that's called the crypt. So stem cells are here, differentiated cells are up here, and these differentiated cells are going to be shedded and they're going to die and leave the tissue. So, how can we actually make sure that there are stem cells that are going to replace the differentiated cells of the intestinal tissue? Well, scientists have been able to do what is called a labeling experiment. They put a label into the stem cell. So you can follow that cell, that labeled cell, in the tissue over time. And that label in this case is a blue marker here at the bottom of these tissue crypts. That labeled cell, now we can follow over time. And we have started to do the labeling in the tissue in a timed way so that we can say after 1 day there's a cell here being labeled. Now I'm going to wait for a number of days, and then I'm going to see that that labeled cell has actually replicated. It has grown into more cells, and these more cells still carry the label. All of the cells that are the descendants of the labeled cell, they still carry the label, and it ultimately gives rise to these ribbons of cells that are going to be ending up here in the remainder of the tissue. So these cells are the descendants from these cells here. That's the conclusion of this experiment. You guys follow me, because this is really important. I label a cell, it is able to divide and give more cells. Yes?
How did you label the cell?
Yeah, so how do we label the cells? That is a genetic trick. This was done in an animal, in the mouse. And you can put into the mouse an extra gene. We call it a transgene. In this case, the transgene is encoding the labeled protein, which is called beta-galactosidase. And by more genetic tricks, we have been able to make sure that that transgene is only going to be functional, expressed in the stem cell. Okay, so now we're going to show you yet another animation, a movie. My own work, because that work has been focusing on the signals, the growth factors that are very important for the stem cells. And these signals are called WNT, we abbreviated WNT in the lab. So these are signals, they're proteins that are able to activate the stem cells to start to proliferate. They come from yet other cells. They're being secreted by one cell, and then they hit the neighboring cell and instruct the neighboring cell to proliferate, to divide. So it turns out that if there's no wind, the stem cells are not going to divide. They're not going to proliferate. So you need the wind protein for the cells to start that whole essential process of cell division. But with wind, they make two cells. So that implies that wind is a growth factor. It is something that is made by one cell type and then is acting on that cell, and that cell is then going to proliferate, make more cells. So then a question that we have been studying in the lab is, how does the wind actually work? What is the mechanism of action by which a stem cell can detect that there's a wind protein outside and then is going to undergo that process of cell division? Well, it turns out that that mechanism includes a molecule called a receptor. This is like an antenna that is sitting on the surface of cells. That receptor is able to recognize the presence of a Wnt protein. And then within the cell, there's what is called a roadblock. And that roadblock is going to tell the cell, hey, do not divide yet. However, when the Wnt protein is now being made and is being presented to the cell with its receptor, the receptor is going to recognize the presence of a Wnt protein outside, and that will inactivate that roadblock. And that will now release the cell from the roadblock activity, and the cell is now going to proliferate. So now you see that a couple of things. You need a signal, a factor, for a cell to become proliferative, to start to divide, and also how that signal is being translated from the surface of the cell to the inside, so the cell are going to proliferate. So now finally I'm going to say a little bit about cancer. It turns out that this whole mechanism of Wnt telling cells to proliferate is tightly linked to cancer. And you can imagine why that is, right? So here's a factor, a signal, that can instruct the cell to proliferate. We also know that cancer is the consequence of cells over-proliferating. They start to proliferate in a situation where they should not. And it turns out that there's a very simple mechanism by which a cell can turn into a cancer cell. Any of you have any guess what that mechanism could be? Talking about that roadblock. I told you about the roadblock. No? The roadblock was inactivated by the signal. Yes?
So like the Wnt signal would stay on there and like that's how the cells would get through?
Would start to proliferate all the time. Yeah, that is one excellent answer to the question, indeed. Because that is actually what is happening in the case of certain cancers of the mammary gland, the breast, in the mouse. But there's another mechanism. And this is a mechanism not where we overactivate something, but where we eliminate something from a cell. We're going to move something out of the cell. We're going to delete it from the cell. So think about a roadblock, yes?
Delete the roadblock itself?
Yes, that's right. So, recall, there was a cell. This is a normal cell, yet still normal. It's going to receive the Wnt through the receptor, and that leads to the inactivation of the roadblock, right? And now the cell is going to divide. Now, we're going to make a cell now where the roadblock is gone. That's right. Yeah. The cell will divide uncontrolled. It doesn't even need the Wnt anymore. It doesn't care that there's no Wnt. It will start to divide all the time. Now this may sound very simple, but that's the way exactly how certain cancers develop. In particular colon cancer, which is quite common, unfortunately. Those are the consequence, those colon cancers, of the loss of those roadblocks from certain cells. So these cells now don't care about signals anymore to the outside, they proliferate all the time. So colon cancer is the consequence of stem cells in the intestine over-proliferating after a mutation. So the mutation removes the roadblock. The gene is called APC, in case you're interested in that special function. So those are quite common in human colon cancer. And many other cancers originate in stem cells where other roadblock genes are mutated. So, I'm telling you a little bit more about myself. I was in a high school, just like you guys are now, in Amsterdam, in the Netherlands. I got my PhD at the University of Amsterdam, Netherlands Cancer Institute, and this is actually, Mark referred to my mentor, Harold Farmers here in the middle. He shares with me a passion for bike riding, which we do all over the world, including in the Netherlands, which you can see by these windmills, right? Everybody can recognize this is from the Netherlands, and this is my brother, just in case you wonder. And this is what we did at the time. We found that the Wnt gene was actually activated in breast cancer in a mouse by a so-called mouse memory tumor virus. And this was the publication that Marek referred to. So, and now this is what I do finally at Stanford, that was already referred to. And now I want to stop the lecture and tell you a little bit about the scientific process. Because if you guys are going to be scientists, you have to understand how science actually works. And this is my spiel on the scientific process. You see something happening. And in this case, what you see happening is that there's a tree that could be growing here on campus, and somebody has decided that the tree has to be cut down, right? Or there must be a lot of beavers here as well. Is that the case in the river? No? Anyway, it doesn't, yeah. But if you watch then the stump, the trunk over time, you see that it will restart the growth of branches and leaves of the original tree. That happens all the time. And if you just look at that, you may think, oh yeah, well, that's the way it is, right? But it actually brings up, if you think about it more, nothing that I ever did in this case, some interesting questions. And the questions are, you wonder what's going on, think of a mechanism, a hypothesis. And one of them could be, so why is it the case that if this trunk had not been cut, that somehow there were cells there at that position in the original tree that had the potential to grow out into a side branch and a leaf, but they didn't. Why is that? Why are there cells here that are only going to grow out into a branch and to leaves, but they don't? And actually also, what is the nature of these cells that have the potential to grow into, a functional tree with all the things that the tree needs, but normally they don't, right? And these cells, indeed, as you probably see the link with my other part of the talk, they're stem cells. They're stem cells in the trunk of a tree that don't grow out unless something has happened. And you can try to isolate those stem cells and see whether you can understand what makes them grow or not in certain conditions. And the other thing that I find fascinating about this situation is that it is not unlikely that these cells that are present here in the trunk of the tree don't grow out because there's somehow an inhibitory effect of the remainder of the intact tree here that inhibits the outgrowth of these cells. Some inhibition. And that is indeed the case. Scientists have later found that mature trees, they secrete all kinds of signals to the remainder of the tree to inhibit the outgrowth of the other cells into side branches. And this is now an example of a day-to-day observation that you can make, that you can test. You can try to isolate a factor from leaves and branches of a tree and see whether that factor is being able to inhibit the proliferation of these cells here. And if you think about other processes that you see from day to day in your own life or in the lab, you can set up a hypothesis that actually tests that can be tested by experiments.